Okay. Hello, ladies and gentlemen. Welcome back to this next lecture, which will give a brief introduction of the neuroscience methods that are commonly used in neuroeconomics. One of the questions that is commonly asked by economists is why do we even investigate the brain? Well, in the end, all of the economic decisions, the decisions involving risk, for instance, decisions involving other people, they're processed by neurons in the brain. So fundamentally, all of these decisions are biological and motivated by biological needs: the need to survive, the need to find food, the need to find shelter, warmth, partners, and also the need to establish homeostasis. So that is, when something is off balance, the brain will try to recreate this balance. So these decisions are driven by biological needs, and they're processed by neurons within the brain. And so this is why eventually the brain is important also for economists. Well, why is this the case? Well, the brain is the seat of our thoughts, our feelings, our desires. And those are obviously things that, when it comes to desires and maybe feelings or emotions, that economists are also interested in. These are things that produce behavior, and that's what enables and constrains our actions. So fundamentally, the brain is involved in behavior and also in economic and social behaviors that we are interested in as economists or as neuroeconomists. And so giving or establishing a broader understanding of the brain will also help us to produce biologically plausible models in economics. And this is one of the goals that that neuroeconomics has. And that will take a few years still. Well, we've been doing this for about 15 years now to have an impact on economic models. It's slowly starting, but it remains an interesting topic. So one of the questions we have in neuroeconomics is, how does the brain enable behaviors? This is, on the one hand, it allows us to make complex decisions. It allows us to plan forward. But also, how does it constrain our behaviors or our actions in a way? So these are questions that we can ask as neuroeconomists by looking at the brain or simultaneously observing neural correlates while people are behaving or making decisions in the scanner or in other types of methods that we use. Here's some facts about the brain. Your brain has about 86 billion neurons, plus minus 8 billion, let's say, for some. And this number, unfortunately, declines as we age. I'm showing a neuron here. Let me switch to the laser pointer. So we'll go into this in more detail a little bit later, but this is the cell body here. Then there's an axon that allows the cell body to communicate its decision in a way to other neurons via these axon terminals. After this point, actually, communication becomes chemical, whereas this communication, sending a pulse down the axon, is electrical. We'll talk about this in a second. Importantly, though, each of these neurons makes about 10,000 connections with other neurons, and this leads to about 860 trillion synapses in the healthy young adult brain. Again, this declines as you become older, but mostly because of efficiencies. And this is what I mean by a synapse here. We'll talk about this again later. This is the process of chemical communication between two neurons, which you learned about in your NeuroAnatomy lecture. So this is a bit of repetition here. Okay, let's look at some methods that are commonly used in neuroeconomics. They can roughly be divided into neuroimaging techniques and brain stimulation techniques. Another way to think about this is to think of them as correlational techniques that observe neural communication while participants are behaving in some way, or manipulation techniques that change the way the brain works, and then the experimenter can look at how this changes behavior afterwards. So in the neuroimaging category, we have positron emission tomography, which basically looks at radioactive tracer isotopes and how they bind to different aspects of brain or neurochemistry. So you can develop a radioactive agent that specifically binds, let's say, to dopamine, and then you can show the dopamine distribution within the brain, or to glucose, and then you can show how glucose changes when the brain uses up the glucose while the neurons are firing. In fMRI, this is a little bit different. We look at the hemodynamic response, which is basically the changes in magnetization as the brain uses up oxygen, or as oxygen gets relocated to different aspects or different parts within the brain. So while they measure relatively similar aspects, both of these methods have wide-ranging applications that can differ quite a bit, and they're very interesting, and they complement each other in many ways. So they're still around. fMRI has overtaken the use of PET, although it's a younger method, but it is so versatile that it can be used for so many different things, and that's why it's used most commonly nowadays. Then we have brain stimulation techniques. They include transcranial magnetic stimulation, which is shown here in this picture. We switch back to the laser pointer right here. This is a TMS coil. It's usually shaped like an 8. This is a bit of a simpler coil, but what it does is it sends out a very strong magnetic pulse into a specific part of the human brain, and if you stimulate in a specific way, what you find is then you exhaust the area under this region, which leads to basically lesioning this area for a short period of time, let's say 30 minutes. So you exhaust the neurons in this area, and they will no longer contribute to the behavior or the decisions that the subject is making for the next 30 minutes, and then you can observe a change in behavior due to this technique. Similar to this is transcranial direct current stimulation, or TDCS, which uses 2 electrodes, an anode and a cathode, and sends electrical signals through this, basically upregulating neurons in one region and downregulating them in another region. And then you can, again, by changing how the brain functions, you can observe how does behavior change in relation to this and make inferences about the causal involvement of these regions in specific aspects of decisions or of economic behavior. TDCS is also widely used to look into other aspects and to treat, for instance, here, stroke or depression. These Italian guys are trying to sell this and find some sort of general aspect of this method that works for all subjects at home. I wouldn't recommend you to try this at home, but you can acquire a TDCS stimulator relatively easy because they're relatively cheap, basically, cost efficient. So those are the 2 brain stimulation techniques. We also have techniques that look at electrophysiology, so exactly how neurons work, specifically at neurons. That is an electroencephalography. You probably know this because that's a very common technique that's been around for almost 100 years, where you put these caps, these bathing caps on people, but they're electrodes placed inside the bathing cap, and then you have these cables coming out of this cap. Here you actually record the responses or the action potentials of neurons, especially at the surface of the skull. So cortical neurons you can record here, but you need about 1,000 firing in unison to be able to pick up the signal there. But you're picking up actual action potentials, actual signals from these neurons. Magnetoencephalography is similar. It's this hairdryer-looking machine that looks at not the electrical properties, but the magnetic properties due to the electrical firing of the neurons. It's a little bit more accurate, but it's also a very sensitive method. And, well, it can be argued whether it adds too much to EEG, and obviously the cost benefit is not very high. There's not many of these around in the world. And they basically pick up the same electrical pulses that electroencephalography picks up. But it is a bit more accurate, and it allows you to do more accurate localization of where the signals come from within the brain, which is one of the problems that affects this methodology. And we have single unit recordings, which is the most direct way to measure electrical activity in awake animals or humans even, where you basically draw, you have to drill a hole into the skull and then stick an electrode into the skull, into the region that you're interested in, and then you record from this region while the animal or the human is making decisions or performing some kind of behavior that you're interested in as the experimenter. So this is a very interesting method that has led to probably some of the deepest insights into how neurons work. And it's a direct way of measuring action potentials directly within the brain. But it's obviously highly intrusive, not like fMRI, where you just put someone in a scanner like this. You have to do this in humans only while participants are undergoing surgery. So most of the time when they're undergoing brain surgery, you wake them up to make sure that you're not affecting their language centers or their motor centers to basically allow them to move and speak after the surgery. When you're taking out, let's say, a tumor or performing other brain surgery, the person is actually awake for a short period of time while their skull is removed. There are no pain receptors in the brain, so that helps. But it's obviously a very awkward moment. And then we have microstimulation, which use these very same electrodes that are implanted in the brain, but not to record activity, but to stimulate neurons within a certain region by sending down an electrical pulse into this region. And this has been shown to change behavior quite clearly. Finally, we have lesion studies that have been performed in animals and humans for quite some time. These are basically opportunistic studies. So when someone had an accident or an infection to the brain, like a virus infection, because viruses also can get into the brain and then destroy some of the brain tissue. We see this in COVID now, where you lose your sense of smell. This is most likely a central nervous system effect of the virus directly on the brain. Or we have seen this with some herpes infections that can travel to the brain and then destroy the amygdala, for instance. And there are also genetic diseases where some people do not develop an amygdala or have lesioned amygdalas or underdeveloped amygdalas. But they allow us basically to study what happens to the human brain in normals compared to patients, or in healthy controls compared to patients, when a certain region is missing or heavily damaged. How does this change behavior relative to healthy controls? And finally, we have animal and human behavioral experiments, which are obviously very important aspects that allow us to inform how the brain works and how the brain produces behavior. And if you combine those with pharmacological treatments, that's an even stronger way of looking at this. Then you can look at neurotransmitter systems by either upregulating them through agonists or downregulating those through antagonists. And then see how does giving a certain drug, let's say a selective serotonin reuptake inhibitor, which increases the amount of serotonin in the brain, how does that affect behavior? Okay, let's do a short review of brain neuroanatomy. It's not too exciting. I know you've talked about it before in some detail in the NeuroAnatomy lecture, but let's go over this briefly before. So we have certain axes in the brain And we will use those when we talk about where the locations are of certain regions, when we do the SPM course, and when you actually look at activations. And to be able to talk about those, we need to remember these terms. So we have rostral and caudal, which lie along this axis here. Rostral is basically the front of the brain, and caudal is basically the back of the brain. This is equivalent to saying anterior and posterior. So we have anterior regions, let's say the anterior medial prefrontal cortex, which would be a region somewhere here. We also have the rostral anterior cingulate cortex. So this is the anterior cingulate cortex here, which wraps around the corpus callosum, which is this white matter fiber tract that connects our left hemisphere to the right hemisphere. And around this wraps the cingulate cortex. And we have an anterior part here and a rostral part as well. But these are naming conventions. They can change, right? So the anterior cingulate cortex is this chunk here, basically. And then down here is the ventromedial prefrontal cortex or orbitofrontal cortex. Again, these terms are used differently across different fields within neuroscience, so it gets a bit confusing. I will mention this when we talk about the ventromedial prefrontal cortex, for instance, in the neuroeconomics lecture. Dorsal also means the top of the brain. Ventral means the bottom of the brain. So ventromedial is down here. One other region that comes up a lot is the ventral striatum, which is about here, also just behind the corpus callosum. But it's better seen in lateral views or coronal slices, which we'll talk about in a second. Then we have superior and inferior. Superior, again, is dorsal or the top of the brain, and inferior is below and at the bottom of the brain, equivalent to ventral. Okay, so I think these terms are quite clear, but have a look at them by yourself again. Then we also have lateral and medial. So the medial prefrontal cortex is somewhere here along this line, whereas the dorsal lateral prefrontal cortex is somewhere around here, right? So those are terms that are common in neuroscience, especially in cognitive neuroscience. So lateral means to the side, medial means to the middle. And when we talk about fMRI, we also have to slice the brain in different directions. And this is something that will become second nature in this course here. So when we look at the brain from the top, this is called an axial slice. So you're basically cutting the brain like this. Obviously, you're not cutting it, but you're cutting the image like this. And you can also record the image like this in the scanner. And you can go from bottom to top. And so this is an axial slice of an MRI image here, for instance, and the axial slice is shown in the green here. And you can take slices from superior to inferior and go up and down like this. Then we have the coronal slice, which is basically slicing the brain from the front to the back or back to the front using this plane here. So basically within the z-axis. And this is how this looks like in the human brain, an MRI image. This is actually a very nice image of the striatum. This is the caudate nucleus here, which is part of the striatum. It's called a striatum because if you look at this closely, this white matter here goes into the gray matter, and it makes these stripes. So striatum stripes. Finally, we have the sagittal plane, which is basically going through the brain in a cut like this from left to right. So basically in the middle, it actually splits the hemispheres into half. Okay, so let me show you a quick example of this, which I can do here with MRICroGL. So this is a software that allows you to view images, brain images in particular. You can add overlays to this with your statistical analyses, but this is just showing the anatomical image of the brain. I just want to show you what I mean by this. So this is the coronal section or coronal slice, and we can I don't know what happened here, but we can scroll through this basically. So I'm going now towards the anterior part of the brain. That's what the A button here means. You can see that the brain changes, and each one of these is a slice. So now I'm in prefrontal cortex, and I'm going further into the anterior prefrontal cortex. I can also go back again, back to the striatum, basically. So we can scroll through the brain like this, and each one of them is a slice that the MRI scanner takes. Then we can go left and right. So now we're working with this sagittal view here. So coronal, sagittal, and axial views. And we can scroll through those. So from the middle of the brain, where you can clearly see the corpus callosum and the ventricle, which is this black area which is filled with cerebrospinal fluid. And then we can go right to the right hemisphere of the brain. You can see the fiber tracts here. This is white matter. This is basically the connections, the axons and dendrites that connect neurons. And now we're in the insula, which is this region here, which is at the very, side of the brain. And the anterior insula is going to be a region we talk about quite a bit in the neuroeconomics course. And then this is like dorsolateral prefrontal cortex here. And we can also go to superior and inferior regions of the axial slice here. So we have the caudate nucleus here. This is the putamen. Again, this is the insula with the anterior part here. That's important. This is probably, depending on how inferior you are, the ventromedial prefrontal cortex already. And then you can scroll through the brain like this here and have a look at these different regions and also see where, let's say you did a statistical analysis, where your activations are, where do your clusters activate. This here again is a ventricle, which is always shown in black. There's no signal there because it's filled with water or fluids, cerebrospinal fluid. It's not just water. It also contains nutrients and toxins that are removed from the brain. So this is just to illustrate how we can scroll through these different regions using MRI images.